Start the recording again.
Okay.
All right. Good evening and good afternoon, everyone. Today we have Professor Justin Cook from the University of California, Merced, who is an associate professor in economics there, and he will walk us through his findings on antibiotics in the context of US and its genetic antecedents and how it breeds resistance subnationally within the US. Justin, thank you so much for joining. We have our standard 40, 45 minutes and 15-minute Q&A, and um, over to you.
All right. Thank you. So just kind of a little background.
Like, share your screen, perhaps, no?
Oh yeah. I have the slides up. I normally do this when I teach, where I give a little spiel before I.
Yeah, sure.
Yeah. Now we're good. Okay. Here we go. So thank you again for having me. This paper is probably going to be like weird, I guess, for what you guys have been seeing. And it's kind of an older paper. I should mention it's co-authored with Jason Fletcher at UW-Madison, University of Wisconsin-Madison. And this paper kind of spills out of conversations that we had when I was a postdoc for Jason like 6-7 years ago. And it's kind of an older paper, but we just recently, and when I say recently, like this summer, we overhauled it. We kind of completely changed almost all aspects of the paper. So feedback at this stage would be excellent. It'd be great to get feedback because we did kind of change how we're doing a lot of the things in it. So the basic idea is like historically, and when I say historically, I mean like before about like the mid 1900s, there really weren't any effective treatments. So I think most of Western medicine was based upon Hippocrates, Galen, these kind of guys. You had to balance the four humors. There's like blood, bile. There's like two different kinds. Anyway, medicine and treatment weren't really effective. There were some ideas about like the transmission of infectious disease, and this is like particularly salient now, especially with the coronavirus and stuff. And I'll kind of talk about these things, the relationships to today a little bit. So people had this idea, there were quarantines historically. We knew kind of maybe people could lead to these infectious events, these kind of pandemics. John Snow in England, with his discovery of like the water pumps and cholera, the kind of founding father of epidemiology. And this was like in the mid 1800s. So there was this kind of idea that there were these hard to see things that were kind of leading to infectious disease. But it wasn't really until like the mid or the late 1800s when Pasteur and Koch kind of discovered germ theory that we kind of really got a good grasp that there are these pathogens that are infecting people and causing these illnesses. And so during the 1918 flu, like this is post-germ theory, but pre-effective medicines. People knew that there were these infectious pathogens around. They knew that there was a flu, but they didn't really have any medicines for it. Okay, and you can see they like masks, which are big now. They wore masks. I was actually reading about it. The masks weren't very effective during the 1918 flu because people would wear them out in public, but when they went inside and around other people, they would take them off. So they had the mask component, but they didn't have the social distancing component that we have today. And then this was the primary treatment for the 1918 flu. It's just aspirin. I think a patent expired for aspirin right around the time of the 1918 flu, so they just kind of said, get some rest, have an aspirin.
Interesting.
So in practice, there were like no personal treatments. Now that's not to say that there weren't like these aggregate health improvements, because once germ theory came around and people knew that there was these pathogens infecting people, there were like major improvements to water quality. And these major improvements to water quality allowed for improvements in like life expectancy and reductions in mortality from waterborne infectious diseases. At the same time, there were these kind of like clearing of swamps. There were a lot of big public works projects that in aggregate had effects on health, but there were no personal treatments. Oh, I forgot to mention too at the beginning, please feel free to interrupt me at any time. I'm used to that and I kind of prefer it, but I'll try to wrap things up in 40 minutes anyway. So the question is, like, in the absence of effective treatments, were there innate differences that led to differences in the virility and kind of the mortality rates associated with infectious diseases? And then if those are present, like if people, due to historical context, do differ in their ability to fight off an infectious disease, were there differential declines in mortality once effective treatment became available? And that's kind of the main question. All right, so this is building off of some work of mine from 2015, where I showed that the transition to agriculture was associated with this large epidemiological transition. That is, like, before agriculture, people weren't exposed to the same diseases as they were after agriculture. So after agriculture, you had large populations. I'll kind of get into this in a little bit. You had large populations, and you had a lot of close contact with domestic animals. This led to a lot of the major infectious diseases that we get vaccinated for today. Like, all of that started after the rise of agriculture. And so the idea of my old paper, which I'll kind of tie in pretty frequently here, is that people that had a longer history of agriculture had a longer exposure to infectious disease, and that kind of shaped the genome in a way that led to resistance to infectious disease, and just broadly infectious disease. And that resistance mechanism is going to be genetic diversity within the set of genes associated with recognizing and disposing of foreign pathogens. And I'll kind of dive into that. Yeah, go ahead.
So my question is, Chirantan here, you just said that this variation in exposure to agriculture resulted in differential genome-based resistance in different populations, but certain regions which got agriculture versus others, it's not really random, right? So agriculture happened in certain places because of some endogenous processes.
Yeah, no, I would say it was the spread of agriculture was endogenous, but the long-term effect on infectious disease may not, would be a little bit more random. And just like, especially, so there's two factors with agriculture. So one is agriculture, and you got to have domestic animals. So there's it's not random, and I'm not going to try to, even today. I mean, I'm very kind of iffy about using causal language. So these are simply correlation type studies. I think the threats to causation are there, but I don't think they're too big a deal. And so I'm not gonna push it one way. I'm not gonna say it's strictly causal. I'm not doing anything too crazy with identification or anything like that.
Cool, thanks.
All right. And so the second paper that, like, I'm gonna combine, like, I'm basically combining ideas from my first paper, and this paper from Jayachandran, which looks at sulfa drugs. The introduction of sulfa drugs in 1937 is like kind of the beginning of an antibiotic era. So sulfa drugs were, like, I think they had something to do with paint, and they tagged bacteria in a way. They were somewhat toxic, but they were also effective. So there were these reductions. Let me just jump to SEMA's findings. So post 1937, there were declines in the maternal mortality rate, influenza and pneumonia, which would be this top right panel, scarlet fever, and meningitis. So sulfa drugs were effective in reducing the mortality rate, like, despite all their problems. And to me, this is just kind of like the beginning of something. So in 1937, sulfa drugs happened. Like, there were these effects shown that reduced the mortality associated with certain bacterial infectious diseases. But then, like, in 1945, we have penicillin, and then we just have this kind of general rollout. So here's kind of a general timeline of antibiotics. All right, so sulfonamides, 1937. In the 1940s, you have beta-lactams, that's penicillin. And then you just kind of have this broad rollout of antibiotics across the mid-20th century. And so what Jason and I originally were doing, we're kind of looking at this very long panel of US states, and I'll kind of talk about that. And we're looking across 1900 to 2000 to look at the broad effect of antibiotics. But then we kind of got some pushback from refs, and they wanted us to focus strictly on the 1937 intervention. And I'll kind of show you results for both. So I'm going to focus today almost primarily on this 1937 introduction date of sulfa drugs, but then I'll extend the panel a little bit, and I'll show you the kind of broader effect of more antibiotics coming into the system. And again, the whole idea is like, okay, so we have these treatments that come online, but do populations of people respond differently from these treatments based upon their ancestral innate resistances to infectious disease? Okay, and so again, like this is going to be a study within the United States. I probably should have mentioned that at the beginning. Essentially, I'm going to create a panel of states that centered around this 1937 intervention date, which is tied to sulfa drugs. And then I extend it a little bit. And I create a genetic resistance score for each US state. Because the genetic resistance score, I talked about this a little bit before, it's diversity, which means it's a population resistance. Let me dive into it a little bit later. I'll just give you the gist of it. So the whole idea behind the genetic resistance score is that within a population, if people are diverse in their immune response, so let's say that my body just produces some kind of different response to an infectious disease, the disease is slower in moving through the population. Okay, so if we're diverse, if there's a lot of diversity within how we respond, then the infectious disease can't infect everyone. So it's not really the resistance mechanism isn't personal. It's basically tied to the population that you're in. Okay, where the diversity is good. Diversity is resistance because diversity actually slows down the pathogen. And you can think about pathogens constantly mutating. So just kind of having differential responses is actually favorable to the spread. And this is a hypothesis by anthropologists and some like medical people, biologists. And it's not, it's shown in animals, it's thought about in humans, but it's not, I think, exactly clear cut that people do have these differences in resistance. So
what is HLA?
Yeah, it's what I'm saying is this isn't like a definitive in science that differences in the HLA diversity within people are associated with differences in health outcomes, even though I've shown it in my past paper. So still,
yep.
In the hard sciences, I don't think this super established. So here's kind of the basic idea of the paper. All right, so I've broken states up into quartiles, and so the circular quartile, I don't know, you can see my mouse. The circular quartile, these are the most susceptible. This is the most susceptible population. So prior to sulfa drugs, they had more mortality from bacterial infections. Okay, but then following the introduction date, they had a larger decline. Okay, and that's essentially what I'm going to estimate in this paper. Can you see my cursor?
Yeah, like we can see your cursor.
Okay.
So just to clarify, Justin, HLA, could you clarify what's HLA?
It's the human. I have a whole set of slides, so I'm going to get into it.
All right.
It's the human leukocyte antigen system. It's basically a set of genes that are responsible for recognizing and disposing of foreign pathogens. There's a large amount of evidence in animals. This is called the MHC, major histocompatibility complex. Mm-hmm. And so in animals, it's kind of well known that this is a diverse set of genes, and diversity is actually good for resistance. In people, it's not as clear, but there are differences, and I'll kind of talk about that a little bit. Okay, so a strong relationship between HLA diversity and pre-1937 mortality rates leads to this differential decline. More susceptible states had a bigger decline. More resistant states had a smaller decline, and that's the paper. It'll be in a different framework, panel of U.S. states for 11 years primary analysis, and then like 70 years extended analysis.
So one quick question, Justin, on that.
Yeah, go ahead.
Can you go back to that slide?
Mm-hmm.
Yeah.
This one?
Yeah, this one. So is this a right way to interpret this result, that for populations which do not have enough resistance, sulfa drugs helped, but those who had net resistance already were being saved from mortality through the through their innate resistance? So like
that's exactly right.
Yes. Okay. And then in that case, what's the normative implication? I mean, can you build innate resistance through some policy interventions, or is it like God gifted?
Yeah, I mean, I'm not going to really touch on policy, but yeah, there could be targeting type policies. I think the idea, and I'll kind of show this a little bit later on, is this, it's primarily people that have ancestry native to the Americas. So
Mm,
people from Eurasia tended to have a lot of exposure to agriculture and infectious disease. People native to the Americas didn't. And so what's going to be driving the results is that states that have a larger population native to the Americas are the ones that are having this bigger decline. And this is counter to what's been shown in the past, first of all. So first of all, a lot of other studies show that minorities in the US tended to benefit less from these treatments.
exactly.
I'm going to show the opposite, that they tend to benefit more. Mostly they focus on black versus white in the US,
Yeah.
but I'm going to say, like, people native to the Americas, like the large Hispanic populations in the Southwest US, they tended to benefit more from these treatments. And so there could be some kind of targeting based on that. I don't want to get, like, it's a little too dystopian for my taste.
Sure,
But I
sure.
mean, I think there should be some kind of at least awareness that, maybe history has shaped responses to, like, our history
Yeah,
has led to differences in the ability of people to kind of differentiate and to fight off infections. And you're seeing this now in coronavirus.
exactly.
I'm not gonna talk about coronavirus too much because I think the data are very preliminary. But there are these big reports that, like, I know in California, like, the infection rate among Hispanic population is like 60%. So 60% of the coronavirus cases in California are among the Hispanic population, and
Mm,
they make up, I don't know, like 20 to 30% of the population. And so, I mean, that could be tied to socioeconomic factors. I'm not going to talk about modern day coronavirus stuff right now. Eventually, I think I will do it once there's data, but
yeah.
there
Okay.
could be just targeting would be my only policy recommendation to shortly answer.
Cool.
Okay.
Thanks.
So again, the primary measure is going to be this population resistance to infectious disease. And so the HLA system is responsive. It's on just 6 chromosomes. It's a set of genes, and their primary function is just kind of how cells kind of interact with each other. So cells kind of bump into each other, and then they're like, oh, they can kind of talk to each other. And so the HLA system is very important in organ transplants. Um, so if you get a kidney from someone, they match you on a lot of these HLA genes. It's like a set of them that they match you on, because again, if you get a kidney from someone that's different than you, your own body may not take to that properly. It's also important. It's like in autoimmune diseases, celiac disease, these kind of things as well. It's just your body not recognizing certain things and kind of fighting it off. And so antigens, human leukocyte antigens, so antigens are proteins on the surface of the cells. And then it's just basically how the cells talk to each other, how they recognize each other. And so if you have diversity, like, my body may not recognize that, I have coronavirus, but, my neighbor's body might. And so I'll get infected, but my neighbor won't. And so that's kind of the whole idea. Um, so yeah, there's some evidence that, uh, this diversity is actually positive within people in resistance to infectious disease. I think the primary thing that kind of stood out to me is, like, if you get measles from a close kin, you're more than twice as likely to die. And the whole idea is that just the particular strain of measles that you would get has adapted to overcome, your kin's immune system. So if you and your kin are very similar in how your immune responses, then the measles strain that was able to infect your kin is going to be also able to, like, infect and spread in you as well. And there's also some preliminary evidence that, HLA diversity is associated with the slow spread of the HIV virus. In mouse studies, where a lot of this stuff comes from, diversity is clearly shown to be a preventative measure to the effectiveness of the viruses and bacteria and stuff. And here's, like, one silly study that I never cite in papers, but I always present it because I think it's interesting. So uh, this Swedish guy, he had men wear shirts for like 3 days without wearing any kind of clones or deodorants or anything like that. And then he asked women to judge the attractiveness of these men based strictly upon the smell of these shirts. And he found the statistically significant effect that women were choosing men as attractive based upon those that had different HLA genes. So this would just kind of propagate diversity. So the whole idea is, like, okay, you want to mate with someone that has a different set of genes than you to kind of create the diversity within the system. And that's what he found by this shirt spelling example. And there were 2 studies that he did. Yeah, so there's one, there's like some website that matches people, like a dating website that matches people based upon their set of HLA genes. Where again, diversity is better. But that's the whole idea, diversity is better. So if you have different set of genes than I do, I'm going to be attracted to you because then our offspring are going to be stronger and they're going to be diverse in regards to their immune responses. Another interesting point about this study too was like, I think if women were on birth control, then the effects were just pretty much washed out. They disappeared, which is just, I don't know how reliable that is. And I'm sure the sample size isn't huge. That's why I don't cite them in my work, but it's anecdotal for a talk. Okay. So where does this diversity score come from? Well, it comes from like 156 different genetic variants. And these genetic variants are tied to 51 distinct ethnicities. And it's from this allele frequency database, which was set up. It's like a repository for anthropologists. So the anthropologists go out into the field, they find some defined ethnic group, and then they kind of genotype them. And then they would deposit those genes at this allele frequency database. And there were 51 of those, which I kind of extend. And the measure of genetic diversity, I think, is somewhat standard now in economics, is expected heterozygosity. This is the probability that 2 people just differ at a particular locus, like the likelihood that they differ at for a particular gene. It's basically a Herfindahl index. It's nothing too complicated. And so one thing that we did in this paper and the kind of new revised paper, and I will apologize that this may lead to confusion, is that instead of looking at expected heterozygosity or diversity, we kind of switched that and we're looking at homozygosity, which is simply 1 minus heterozygosity. So I'm going to use terms like similarity and susceptibility, and that just kind of frames the analysis in a way that I think is easier for others to understand, but not for me. So I may be a little, because I kind of grew up thinking about like, okay, diversity is good, diversity is, now I'm kind of switching. We're going to look at the effect on being more susceptible, least resistant populations. Okay, so expected heterozygosity is calculated at the ethnic level. Ethnic data are then aggregated to country based upon the ethnic composition of the country, and by matching those 51 ethnicities that have data on to other ethnicities based upon language groups. Okay, so if you're, I think French as an ethnicity, so like people in Belgium would be assigned to French, like at least half of them. So it'd just be based upon like language classifications. Okay, so getting into the origins of this, so I kind of touched on this earlier. So why do people differ in their resistance mechanisms? So one is going to be out of Africa migratory distance. So the Oshkosh Glory paper showed that people that are further along these migratory patterns out of Africa are going to have less overall genetic diversity within, they're just going to have overall less genetic diversity. So as we move along these migration routes out of East Africa, you get less and less genetic diversity. And this is due to serial founder effects. The idea there is that a smaller subset of the population is moving out each time. And so that subset only contains a fraction of the total amount of genes. And so you get less and less genetic diversity as we move along these migration routes out of East Africa. And that's overall genetic diversity, but it's also going to tie to HLA diversity. And a key point here is that out of Africa migratory distance, if you're just looking at kind of the residual, it explains 80% of the variance of overall genetic diversity. But out of Africa migratory distance only explains like 17 to 39% of HLA diversity. So something else was happening with HLA diversity. That's kind of my main point. It's like, okay, yeah, so it's tied to out of Africa decisions, but it's also being shaped by the ancestral world. And so you can see, here's just kind of a simple plot of migratory distance from East Africa and HLA diversity. And European countries tend to kind of cluster up here above the line, above the fit line. Asian countries are kind of here as well. And African countries, Africa has the highest amount of genetic diversity, are fairly high as well. So the second kind of explanation as to why people differ in their resistance is like agriculture. So I mentioned this before, is like, so once people switched from like hunter gathering into agriculture, there were two main effects of that. One, they had like large dense populations. Large dense populations accelerated the spread of infectious disease. The second is they had access to domestic animals. And so this is like some Jared Diamond stuff here, but like if you had access to domestic animals, then you had a lot of close contact with the animals. And then you had these zoonotic episodes, which I think we're all somewhat familiar with now, where the pathogens would jump out of their animal hosts and into humans. And that's how we got a lot of the infectious disease we have today, like measles, mumps, rubella, like all those things come from domestic animals. I think measles is cows. There's a list. And these are old slides that I'm kind of recycling here. This one over here on the right, this is a market, one of these open air markets in Guangdong, China. And so this is where they think, I initially put it because this is where SARS came from, but I think somewhere similar is kind of the coronavirus. So it's just kind of these zoonotic episodes happen historically, and they happen kind of irregularly, and they were tied to like contact to domestic animals. And here's where I just showed this, that the genetic diversity does have statistical associations with these preliminary causes. So just years of agriculture, more years of agriculture, more diversity within the HLA system, strongly associated. Further out of Africa, less diversity. So further out of Africa, less diversity, more years of agriculture, more diversity. Domestic animals, positive association. And then population densities, sort of population densities also accelerate the spread of infectious disease.
Why is N 131?
Well, I'm sorry, what?
Why is N 131?
Oh, this is from my 2015 paper. So this is a cross country study. So that's the max number of countries I had for that particular sample.
Okay.
Yeah. And this is cross country and country level, and this is from the old paper. So I just want to show that the statistical associations were there in a way that I've been saying. All right, so what are we doing for this paper? So we're going to look at U.S. states. We're going to look at the contiguous U.S. states, so 48 states. I have this data on ethnicity and country level, HLA diversity or homozygosity. And so we want to convert that to the U.S. state data. And so how that's done is I use census. I use the 5% census sample for 3 years, 1980, 1990, 2000. 1980 is the first year in which people are asked about their ancestry. It happens in those other two 5% census samples as well. Okay. So people are asked about their ancestry, they put that down. And then I match that reported ancestry to either a country or ethnicity for which we have data. And also I'm trying to backtrack, I'm trying to get things prior to the treatment date, 1937. So I use state of birth, so the reported state of birth for the person, and the 5 years prior to 1937, so 1932 to 1936. Okay? So it kind of creates these representative state cohorts and then their ancestries for the period prior to the treatment that I'm going to look at, which is 1937. Okay? And then I match those reported ancestries of that kind of pre-1937 cohort to countries and ethnicities for which I have data on HLA heterozygosity or HLA homozygosity. Okay. And so this is kind of what it looks like. So there's just kind of, a heat map of where diversity is. So the lighter areas are going to be more susceptible. Arizona in particular is going to be my most susceptible state. I'm going to talk about that. The Southwest is going to be more susceptible.
So Justin, this is Anindra
yeah,
here. Go ahead. There would be a pretty large standard error for this kind of backcasting techniques, right? Can you explain a bit more about how you created the sample before 1937? Because I think you're showing the mean estimate here.
Yeah.
That would also
I'm going to use the mean. Yeah.
Error.
The standard error would be big.
Yeah. I mean, maybe I couldn't quite exactly follow how you created the sample. Can you please explain it once more?
Yeah. So it's an aggregation. So it creates these kind of representative populations for those born in a particular state within the 5-year band, the 5-year window before 1937. So there'll be like, you know, it's a 5% census sample, so it's like thousands of people per state. It's not a ton. And that creates ethnic compositions of the state. So it may be like 50% British, 30% French, 20% Black, something like that.
And the assumption here is that that is close to a random sample.
I don't think I can say it's yeah, so the census sample should be a random sample. There could be some selection into who's actually making it.
Right.
So some of those people may have died. There could be selection in that way. But yeah, I would say it's this, because it's based on the census sample, it's close to random. The idea, there's a concern of selection where that certain individuals may have died between their birth in 1933, let's say, and 1980 when they're first recorded in the census. And that could have been non-random.
No, I mean selection in the sense that, yeah, that is a possibility. The other possibility I was thinking is that you are essentially backtracking from the people who have reported it in around, you know, much later, right?
Mm-hmm. Yeah.
So if there is a self-selection in terms of who are reporting in, let's say, close to 2000, then that selection bias also goes back to, you know, constructing.
Yeah, so I'm going to lean on the census here, the 5%. I think the census people are doing a good job of randomizing the
okay, all right,
sample at that point. Yeah, the concern that the concern, and I kind of talk about this in the paper a little bit. There's one concern that like maybe particular groups of people are just dying off at greater rates, and they're not making it to 1980, 1990, 2000. And then that's coloring my historic diversity score. But I think the direction of bias in that case is going to push our estimates downward, because what you would have is you'd have higher mortality in these particular states, and you'd have lesser diversity. And the reason is because is like if the more susceptible populations were to die off, that diversity wouldn't be shown, and that lower level of diversity wouldn't be shown, and you would have higher mortality. And that's going to be the opposite of the opposite of the relationship that I'm going to kind of estimate. I don't know if that's exactly clear, but hopefully I'll bring that point up again and clarify things when I have estimates and stuff. Okay. So yeah, most susceptible states are states that generally have high Native American ancestry. Arizona, New Mexico, Texas. Mississippi has a large Black population, which is, I'll talk about how I construct this measure in just a little bit. Well, I think next slide. Okay, so one thing I forgot to mention is that how this is being constructed too is I take the diversity score of the particular ethnicity, and then each state is just a weighted average of these different ethnic diversity scores, okay? And so what this assumes is that these ethnic groups are essentially segregated within state. So it's like, okay, so your state's 60% white, 40% Black, then it would be 60% on the white diversity score and 40% on the Black diversity score, and that's how I'd get the state diversity score. And the assumption there is that the populations are segregated and that infectious disease networks and all these things would also be segregated. And I do not think that's right. However, I do think it's closer to the actual state of the world at that time. But we can test this. So the primary measure considered is ethnic groups to be segregated, but an alternative measure can be constructed in which we consider the ethnic groups to be mixed. So instead of that 60%, here's your diversity score, 40%, here's your diversity score, and that leads to a state score, what we can do is we can say 60%, instead of weighting the diversity scores, I can weight the genes. So I can look at the gene frequencies across the different ethnic groups and create an aggregate kind of population that's a weighted average of the gene frequencies, and then create a diversity score from that gene frequencies. And I know that's not terribly clear, but the whole idea is that we create two bounds. We create one bound in which we consider people to be completely segregated, and we consider another bound in which we can create a score in which people are completely mixed. And so we think the true state of the world lies somewhere in the middle of there. I think it lies closer to the segregated side, especially in 1937. But we test both effects and we find very similar results. And I'll kind of, I'll show you those as we go along. Oh, and the mixed spore is going to be like heavily tied to like the Black population in the 1930s. So the South, they're going to have much higher diversity spores with the mixed spore because they have much more diverse populations, because they, you know, instead of being 80% white like states in the Midwest, they will be like 50% white, 50% Black. And so our effects are tied to like controlling for regional differences. And okay, so here's like the mixed score where I talk about, this is where we consider population completely mixed. So Arizona and New Mexico still remain the most susceptible, but then like white states, states that have lower amounts of diversity, like just ethnic diversity, are going to show up as well. So North Dakota, Minnesota, Wisconsin, Midwest states that, you know, are pretty much uniform. Wisconsin's like pretty much 80% German at this time. Minnesota's probably something similar. I don't think they're German, maybe Swedish or something. And then the least susceptible states, so states with the most diversity, would be Louisiana. Louisiana will be the most diverse. So this is a state in the South, a lot of, you know, at least ethnic kind of diversity here, where it's 50% white, 50% Black. Maybe not exactly that, but something similar. Florida, North Carolina, it's all going to be southern states. They're going to have the most diversity with the mixed score. Okay, real quick rundown of the outcomes. I'm going to look at the mortality rate from bacterial infections. I get this from the National Vital Statistics. I consider basically, like, I think it's nine infectious diseases for this bacterial infection mortality rate. Typhoid, scarlet fever, syphilis, maternal mortality, which is a proxy for parochial fever or something, whooping cough, tuberculosis, diphtheria, influenza, and pneumonia. Influenza is caused by a virus, but it's always coupled with pneumonia. And pneumonia could have bacterial causes, and diarrhea and enteritis. We center the panel of states, so we center the data around the 1937 date. We do 5 years post and 5 years prior. So we create a panel from 1932 to 1942 as our primary panel. But we also go longer. The original version of the paper, we considered a 100-year panel. We considered 1900 to 2000. We're going to use a shorter but still extended version of that panel. We'll use 1900 to 1930 or 1970. The problem is that in the early years, so 1900, 1901, et cetera, you don't have all states in the panel. Not all states report biostatistics data in the US until, I think, 1933. Yeah, 1933. We'll also examine as a placebo test residual mortality, which would just be the state's overall mortality rate, less out the bacterial mortality rate. So it's just the all-cause mortality rate minus the bacterial mortality rate. And this is used as a placebo test. The idea is that the treatment is affecting only bacterial causes, and the intensity of that treatment is only going to be associated with changes in bacterial mortality rates. Some controls, I may need to come back to this as when I show you the tables. It's basically going to be rain and temperature. And then I'm going to do a number of different, so the percent Black, the percent foreign born, urbanization, ethnic fractionalization, migrants, and the initial population. So these are time invariant. And so how those are going to be entered into the estimating equation is that I'm going to estimate the post-1937 difference within the panel. So in a similar way to how HLA susceptibility is entering into the panel. I also look at some infrastructure controls like schools per square mile, like hospitals, doctors, and initial income. And then we'll use state and year fixed effects. And our primary measure uses year by census division, which are 9 subdivisions within the US. Okay, so here's kind of what I showed you before. And here's just plotting this by means. So we just break up states into quartiles tied to their HLA susceptibility, where the circles are going to be the most susceptible, the least resistant, and the triangles are going to be the least susceptible, most resistant. What you see is like, here's 1937 intervention date. All of them kind of decline. So bacterial mortality rates decline post 1937, but this decline is going to be steeper for those that were more susceptible. And eventually I can show you this out to 1970. These converge. So that's the whole idea of the paper is like, okay, the most susceptible populations, they're having a more rapid decline and they're converging to some kind of post treatment mean that all states kind of converge to.
Can I ask a question here?
Yeah.
So I was thinking, looking at this figure, it looks like the fall has started at 1936 rather than 1937.
See, I was thinking 1938 myself. Like, this is where the kind of decline happens.
No, I
But
mean,
what
if
do you think?
you move this, this vertical line to 1936, you can
Yeah.
pretty much see that each of these categories are essentially going down in mortality, right?
In 1936? It seems like they're
Yeah.
going up.
No. I mean, after 1936.
Yeah, so that's 1937, where the line is, like, that's the actual treatment date. That's the one proposed by the Jayachandran and all paper.
Oh, okay, so that's not 1938. Okay. So
Yeah, I mean, so I'm kind of accepting that. I'm basically going to estimate this whole thing's going to be our post.
yeah, sure.
1937
Okay.
on is going to be our post. So this is the pre-period. And then I do this, I extend the samples too. So it's not something where I'm just looking at a very selected time frame. I'll extend it by, like, 30 years on both sides. Here's that same kind of relationship, but here's the residual mortality rate. So again, this is all-cause mortality, taking out the bacterial mortality rate. And you can see that the relationship kind of completely flips, and then there's no real difference from the post-1937 treatment. It's just kind of, things are the same, and then the relationships flip. And essentially, I'll show there's no relationship with the residual mortality rate. So other causes of death aren't being affected by either the treatment or the intensity. Okay, so this is like a gene-environment interaction, which is something that the genoeconomics literature is a little bit bigger on. The whole idea is, like, you have this treatment come on, 1937, we have this intensity of treatment, so each state has a propensity or a susceptibility to infectious disease. And so those states that are more susceptible have a greater decline. Those states that are less susceptible have a smaller decline, as has been said a number of times. HLA susceptibility, that's going to be the intensity of the treatment measure. And then we will use 1937 as the intervention date for all states to avoid endogenous take-up, or et cetera. Okay. So 1937 is going to be the treatment date for all states, and then the response is going to be based upon their HLA susceptibility. And again, the idea there is less diverse, more susceptible. More diverse, less susceptible. Here's our base estimating equation. So we will examine, so our outcomes are going to be the bacterial mortality rate, so that's bacterial deaths per 100,000, I think, and the overall mortality rate. So that's going to be YRT. I do standardize HLA susceptibility so that a 1 standard deviation increase would, the coefficient beta 1 is just going to measure the effect of a 1 standard deviation increase. So HLA susceptibility is standardized to a mean of 0, standard deviation of 1. And then we have, like, our climate time varying controls, and we have our time invariant controls, which are also interacted with the post-1937 indicator. And then state and year fixed effects, and mostly year by division, census division fixed effects. Okay, so here's kind of our main finding. I'm kind of running a little late too. So the whole idea, so a 1 standard deviation increase in susceptibility for a particular state is associated with about 15 to 17 fewer deaths per year, bacterial deaths per year. Okay. So here's kind of a piecemeal introduction of controls across our 1932-1942 panel, columns 1 through 5. So there's about a 14 to 17 person fewer deaths for more HLA susceptible states. And so they had larger declines in their bacterial mortality rate, as I've been saying. And the coefficient's fairly stable throughout the introduction of these controls. If we go longer, the longer panel, column 6 here, this is 1900 to 1970, the effect just gets bigger. Okay, because there's been a steeper decline. You can see, like, just looking at the means, the mean bacterial rate is going to be smaller because we have a longer panel, but the pre-period, like, you get much more bacterial death when you include years prior to 1932. And then the post-1937 years are going to lead to a much smaller mortality rate, mean mortality rate. And here's how like the coefficients plot over time. So here's an event figure where I'm just taking the relative coefficients to the year prior to treatment, which is 1936. So prior to 1936, there's no differential effect in HLA susceptibility's association with the bacterial mortality rate. Okay. So our pre-trends look okay. Following 1936, however, you have this kind of blip up 1937, but then you have consistent negative coefficients, which were estimated here. So this is where our negative coefficients coming from. And it's growing over time. If we were to look at the effect on the residual mortality rate, which is again the all-cause mortality rate minus bacterial infections, there's no significant effects. The point estimate of the coefficient is roughly similar size, but I want to kind of warn against that because the means are much bigger. So the mean residual mortality rate is, 4 to 5 times, about 4 times what it is for the bacterial mortality rate. And so there's no significant effect. Here's the event figure for that. So just the relative 1936, how do the effects of HLA susceptibility look? And there's no post effect. There's some noisy, I think 1934 pops up as significant. That's just a relative difference. If I look at the year by year, so HLA association with residual mortality in 1934 and 1936, they're both insignificant, but they are significantly different from one another. That's why this 1934 pops up. But again, following the treatment in 1937, there's no association. So it seems like our effect is working strictly on bacterial mortality rate. Here's the mixed score, which again considers all populations mixed, and this kind of it assumes that ethnic groups are completely like mixed into a giant kind of representative ethnic group at the state level. And so what you see is like if I don't control for census region effects, the effect is insignificant. And that's primarily due to the South having more bacterial mortality, as well as being less susceptible. But then once I account for the regional effects, the coefficient basically becomes similar in magnitude to the segregated score. So I don't think anything too crazy is happening with the kind of ethnic makeup. And so again, it's just more susceptible states had 14 fewer bacterial deaths per year.
Justin, can I ask a question?
Yes, please do.
This, my worry is that the unit of observation is at the state time level, whereas the mechanism is operating through an individual's exposure to genetic diversity. So in an ideal world, if everything was available, you should have done like individual state time regressions, no? And controlled for some sort of unobserved heterogeneity an individual may have to a state, and with time, and then see whether these results pan out still the way you are seeing it. I don't know what I'm getting at, but basically I'm a little worried about the mechanism perhaps here coming from the underlying population effect. Yeah, go ahead. You had to say something.
I don't have individual data like that are suitable for this. So there's a data constraint here because I do think it's a population difference too. So I think it's an aggregate difference. So I think using the state is okay because it's just saying that some states have more resistance, some states have less resistance. So the resistance has to be calculated for a population.
Okay, that's the assumption we started.
Yeah, so it has to be of a population. And then, so the mechanism, I try to control for relevant factors. So the whole point is this mechanism. Like it's just that some states had innate resistance, some states did not have innate resistance. Okay, and I'm showing that my assumed measure of innate resistance is associated with declines how I would expect. These less resistant states are having bigger declines.
Okay. Yeah. Don't worry about it. Yeah, I got you.
Yeah,
Okay.
I mean, I'm kind of, I think it needs to be at a population because there is segregation that needs to take place because the diversity scores like there are based on populations. But it would be if I had individual data that were tracking individuals over time, and I could see if an individual lived in a more susceptible or least susceptible state, and then whether they died, like I think that'd be great. I could do it, but I don't think the data exists for that. All I have are these kind of aggregate statistics.
So actually,
So
there is a
my unit of
question.
observation, 8-year
Yeah.
level. Yeah.
Yeah. So there is a question that somebody has shared, Gaurang has shared privately with me, and this relates to the individual discussions that you're having. He says, in healthcare insurance scenario where information asymmetry exists between patients and the agency, whoever has better idea about possible likelihood has an upper hand. So in general, insurance firms have better chance of getting their hands on gene data and therefore make inferences basis that. Question is which part of the technical process then needs to be made cheaper so that one could make it more accessible and feasible from the patient side and information asymmetry can be tamed. So is 23andMe kind of data helpful then? I don't know. I mean, how do you resolve it?
I mean, yeah, I think it would be. I would need to aggregate. So I would need to create a group, like, and that could be like county. It could just be anything. Like it could be a social network, whatever. 23andMe data would be great. I don't have access to that. It would. Yeah. So another kind of related idea I've thought about doing with this stuff is like nursing homes. Like, so I think there
Yeah.
is some kind of. So if you just look at within the nursing home, within closed environment, the flu and pneumonia kill a lot of senior citizens, and coronavirus now too. But if you were to look at the amount of diversity within those nursing homes had more diversity, would be better off. And that kind of
Right.
idea is kind of what I'm doing here, but at a state level. Just states that have a lot more diversity tend to be doing better off prior to the treatment, though. But I can look at doing in some insurance data or something like that. And 23andMe, I think really the coronavirus pandemic has opened up, like, guys, I was like, okay, I mean, infectious diseases don't really have, at least from my knowledge, they don't have these broad geographic differences. They probably do, but it's tied to like income. So income, conditional income, are they these broad differences in mortality rates? And I just didn't think they existed anymore, but I think that now that we're in a new world, a worse world, that may be the case. But yeah, I mean, that would be a cool extension. So
Cool. Yeah. Go ahead.
here, let me get back on. Let me finish this up because I'm running out of time here. So the mixed mortality rate, same kind of effect. Pre-periods look okay. There's a post-decline. That's where it's coming from. 1937 still blips up. The mixed measure also has an insignificant association with the residual mortality rate. So it's not associated with, like, just death. It is tied to these bacterial causes, as I've been saying. So one concern I had was like the white versus non-white. Like, is this driven by primarily white states? And you can see white states tend to have more. Those states with European ancestors can have more resistance. This is the Europe fraction relative to out of Africa migration. And you can see, like, here's just the correlation between like the fraction ancestral to Europe. So I'm using those reported ethnicities from the census, and I'm just assigning those to a particular continent. And so people from Europe are less susceptible. There's a pretty strong correlation, 0.85. People to the Americas, the flip side of that, are more susceptible. So if a state has a greater fraction of people native to the Americas, then they have a higher susceptibility score. And that's where the segregated. For the mix, the Europe kind of component drops out for the mixed score. And basically you get the black fractions, or the fractions ancestral to Africa. If you have more black, a higher black percentage, then you have less susceptibility. And again, but the Americas still holds up.
So here's my other concern, Justin, which is about omitted variable stuff. So there's so much happening by state time level during this period. Like Marcella Alsan's work on Tuskegee, right? And medical technology is changing. Maybe some observed controls of exposure by state time to this other stuff. Can you bring that in to make this a little more robust?
Yeah, I think that's a good point. I've been a little lazy and just kind of not collecting state time controls. I would mention that I am controlling for the percent of the population that's Black in 1936, as well as a number of other like pre-period kind of like demographic controls. The idea there is that, you know, the response, the post-1937 response may be tied to these underlying demographics. So I think that would get at that a little bit, but having the state time would be better than having just the static control that I'm using.
Yeah. Okay.
So then the final thing I did, another question, or you mean?
No, go ahead, finish it up.
Yeah, so I just split the sample by the European fraction. So I'm like, okay, let's just, you know, just split it. We'll go above and below median European fraction. We'll see how the effects look. And so if you look, you know, below median European ancestry, the effects get much larger. So just looking at states that are beneath, that have smaller white fractions, you get much larger effects in the decline in bacterial mortality tied to HLA susceptibility. So instead of like 14 to 17 fewer deaths per year, you're getting like 36. Okay? So the effect is basically doubling. That also holds true for the longer panel. So that's why I get the bigger coefficient here is longer panel. You probably don't remember, but it was like 66, I think, in the first table. And so the 36 number holds for both the segregated measure and the mixed measure. Looking at those states that are more white, so this would be like the Midwest, the Northeast, the effect of HLA susceptibility is similar in magnitude, but it becomes noisy with a smaller sample size. It does become slightly significant when looking at the longer term. So just within that set of 24 states. The mixed measure does hold though, similar significant effects. Okay. So just to kind of wrap up here, because I have like 2 minutes left, it appears that this innate measure is tied to the response of states from the introduction of sulfa drugs in 1937. And then there's even bigger effects if we look at a longer panel and consider the full breadth of antibiotics that come online in the mid-20th century. States that were more resistant had lower mortality rates prior to the treatment, and they weren't as affected as much from the treatment. So this would be like, mostly European, states with high European ancestry. The opposite is true for states that had, high Native American ancestry. They tended to have, they tended to be more susceptible, have higher mortality before 1937. And then once the treatment happens in 1937, they have this steeper decline. So they're kind of benefiting more from the treatment effects. Yeah, and going further, I thought this was, my last paper on HLA diversity and these kind of things. Aside from, I've thought about doing the retirement home stuff, but I don't know how much, I don't know if I could get data for that relatively easy. But now with COVID-19, I think I may see if this relationship holds both across countries and within US states and counties and these kind of things. Because we do have a contemporary novel infectious disease. And so HLA diversity within your local network should provide some kind of resistance conditional upon the virus being equally accessible. So that's I kind of been struggling with the obvious, I haven't written a COVID-19 paper. Most everyone's written a COVID-19 paper. Most economists, it feels like, at least in my department. I haven't written one. I'm waiting for data to kind of clear up a little bit. But I think there are obvious ties, this should transcend. So your local network, the amount of diversity within your immune response genes should be tied to the spread of this novel pathogen within that network. And I kind of want to see if I can do that in some way.
So I basically remembered reaching out to you, thinking about your work on this innate genetic resistance, because the way it's evolving in India, non-pharmaceutical interventions are essentially falling through the cracks. There are no stable preferences on pharmaceutical interventions. We don't know when the vaccine will come. And ultimately, policymaker is relying on like a steady state genetic resistance, potentially heterogeneous across regions. So
yeah, I think the U.S. is doing something very similar. So I guess they may have access to farmers. I don't know.
is there a global database that captures variation in these HLA genes apart from
Is there,
a
is there what?
global database of these genes?
Yeah, the problem is that what I did is effectively global, but it's based upon these 51 underlying ethnicities, which is problematic. I think you'd want more groups. It's
This is
also,
the
the Alfred, a low frequency data. The problem is too, it's like, I think what you want, I think the better measure would be like your local network, and so you can have like even like, so ethnicities are kind of making assumptions, and they're like a proxy for what you expect the gene frequencies to be, but that may not be the case. Like there's still variation. And so what you'd want is like your kind of local environment, the amount of diversity in that, like the people that you would interact with. And the idea there is more diversity in that environment is going to be beneficial. And so, yeah, kind of tying this to modern day stuff, I think is going to be a challenge for a number of reasons.
All right, awesome presentation. Thank you so much, Justin. I think there are some comments which also praise the presentation, and it's very timely. I wish you luck on your first COVID-19 paper. And if there are residual questions,
if I write one.
Yeah. If there are residual questions from others, please unmute yourself and ask, or else we'll wrap up. Anindo, if you have questions, Mayank. All right, looks like we are good. Thank you so much, Justin. Please stay safe and take good care, and hopefully we can have you physically in campus.
Yeah, that'd be great. That'd be fantastic. I have written, coincidentally, I've written a number of papers on India. I know nothing about India, but I have 2 papers on India right now.
Yeah.
I have one examining in Rega, and then I have one on the demonetization event.
Yep.
So
we need to have you here.
Please. Yeah, it'd be great. It'd be great because again, I'm pretty ignorant on most things, and so I've never been to India. It'd be awesome.
Super.
So thank you for having me too.
My pleasure. Take care, Justin. Thank you so much. Thanks, everyone.
Yeah, thank you everyone for having me, and thank you for the comments.
Bye-bye.
Okay, bye.